Okay guys, so today we're going to go ahead and go over Chapter 5, Part 3, Nucleic Acids. You have this. Please fill out the columns for all biomolecules when you're finished with your notes. So let's go ahead and go over this. So what's the function of nucleic acids? Nucleic acids store genetic information and pass it from parent to offspring. Segments of DNA called genes contain the instructions for synthesizing proteins. So this is a processing piece. Label each molecule shown below. We'll also go over this inside class. Just kidding. Here's the labels. Let's go ahead and put the labels. Okay, moving on. What are the monomers of nucleic acid? So nucleic acids are made of monomers called nucleotides. Nucleotides have three parts. They have a sugar part over here. They have a phosphate, which is this entire group right here. And they also have a nitrogenous base. Two categories of nitrogenous bases are purines. They're bigger because, they're just bigger. So a trick to remember it is that angels are pure. So this is adenine and guanine. And then our second group is pyrimidines, and they're smaller. They are cytosine, thymine, and uracil. To me, it seems like they spell the word cut. All right, let's go ahead and move on. So there's two types of nucleic acid. We have deoxyribonucleic acid, which is DNA, and then we have ribonucleic acid, which is RNA. So the three types of RNA are mRNA, tRNA, and rRNA. We do have some differences between DNA and RNA. So we have RNA over here, and this side is DNA. RNA is single-stranded. It includes AUGC. The sugar is actually ribose. It's less stable, and the three types are mRNA, tRNA, and rRNA. For DNA, it is double-stranded. It's a double helix. It includes AT, G, and C. The sugar is deoxyribose, so you can see the difference right here. It's missing an oxygen. And it's also more stable. Now I know this says draw a picture illustrating the differences between RNA and DNA, but I actually put that picture in your notes, so we can skip that. So how are nucleotides joined together? Can you identify the three parts of these nucleotides? And which are the parts that you think will be joined together? Well, remember, it's either going to be dehydration synthesis or it's going to be hydrolysis. So they are joined together by dehydration synthesis. This forms a phosphodiester bond or covalent bonds between the hydroxyl group of the 3-carbon of one nucleotide onto the 5-carbon onto the next. So we have this phosphodiester bond right here. Why is it phosphodiester? Because I have a phosphate, and on these sides, I have an ester. So an ester is an oxygen in between, and I have an oxygen right here. So phosphate, diester, di means two, and I have two ester bonds. And why are they considered ester bonds? Because it's an oxygen that's connecting the two. This creates a sugar phosphate backbone. We're going to go ahead and go into this in detail in class, and we'll even make a little candy DNA. Yummy, yummy. So if you can, try and circle the nucleotides and indicate which end of the DNA strand has the 3 end and which has a 5 end carbon. So what is the structure of DNA? Well, it's actually anti-parallel. In DNA, the two backbones run in opposite 5 to 3 directions. It's actually called a double helix, which we've also learned in 9th and 7th grade. There's complementary base pairing, which is where our nitrogenous bases are held together by hydrogen bonds. A pairs with T or U for RNA. T or U for RNA pairs with A. C pairs with G, and G pairs with C. Go ahead and try and write the DNA complementary strand to this. And then go ahead and write the mRNA strand that would be complementary to this DNA strand. Try and write that, and then we'll go over that in class. Well, let's go ahead and move on. So our processing piece says to go ahead and draw a sketch of a DNA molecule. Label the sugar phosphate backbone, the 3 and 5N, the nitrogenous bases, 1 nucleotide, hydrogen bonds, and phosphodiester covalent bonds. We're of course going to go ahead and do this in class. If you'd like to practice, you're more than welcome to, but we will be doing this in class. Our processing pieces. The nitrogenous bases in a sample of DNA are found to be 40% A. What is the percentage of the other 3 nitrogenous bases? Well, if I think about it this way, 40% is A, which means another 40% would have to be T because it matches up. So 40 plus 40 is 80. That means I only have 20% left, but I have to split it between these two. So 20 divided by 2 would be 10% each. So in case you kind of got a little bit lost on that example, we'll be going over this one, and I want to let you try this second example by yourself in class. So again, like I said, if you're a little bit lost on this, this is actually a processing piece that we're going over together in class. So we have semi-conservative replication. So if we're looking at semi-conservative, I want you to look at the blue. Are both of them completely blue? I can see that blue is on both sides. So it seems like the old strands are in both of the new strands. And the new strands have this orange side that is added. So let me say that again. Your original strand is blue. When you make a copy, the new strands are added, which are orange. So the new strands are half new and half old. So how does DNA replicate in the first place? Well, DNA replication is semi-conservative. DNA strands split apart. One strand is used as the template to synthesize a new strand, and then the new DNA molecule contains one old strand and one new strand. So we're going to go ahead and do this processing piece tomorrow together. Let's talk about the steps of protein synthesis now that we're talking about protein. So you have this diagram, and I want you to go ahead and fill everything in. So step 1, I have transcription. Transcription is DNA turning into mRNA inside the nucleus. Number 2, I have mRNA leaving through a nuclear pore. So here's the mRNA. Now it's in the cytoplasm. Step 3, I have translation. Translation is when we have mRNA that is synthesized using a ribosome to go ahead and make a polypeptide. And here's that primary structure that we were talking about in that last video. These are all monomers that are waiting to be put together to this polypeptide. So the central dogma of biology is DNA is transcribed to mRNA, which is translated into a protein. So we are going to go ahead and go over this processing piece a little bit more, especially this diagram. This is very important for AP Bio. In the meantime, don't forget to go ahead and go over, let me go back to this beginning part. Don't forget to go over this right here and fill out this entire chart, because by this point, we've already gone over all of the biomolecules. Thank you.